to introduce Dr. Bette Korber. If you joined early, you know that Bette and I have known each other for a very long time. We were grad students together, and her expertise, she's run a group at Los Alamos that's built up the HIV database for the world, and she has been instrumental in really getting to the point where now we have an HIV vaccine that's out being tested, and it really was the work of her group that enabled so much of that. At a more personal note, it's really a great honor that among all the other awards that Bette's had, is she received the Richard Feynman Award for Innovation, because I think Dick Feynman is a hero to many of us that were at Caltech at the time when he was still there. So what Bette's going to talk to us today about is something that's actually in some ways very related to her work in HIV, but of course looking at SARS-CoV-2, and that's tracking the evolution of variants and how that actually informs us about the biology of SARS-CoV-2 and how that impacts COVID-19, the disease. So Bette, if you're ready, we will
I'm ready.
Take it away from here. Usman,
I'll stop
you're
sharing.
controlling the screens. Okay.
Yeah.
There we go.
Bette, you should be able to share now.
Okay, thank you. Let's see.
And welcome, Bette. It's great to have you here.
I appreciate the opportunity to share ideas with you guys. So can you all see my slides?
Yes, we can.
Great. Okay. So basically, I'm going to talk to you about the recent evolution of SARS-CoV-2 variants. I'm going to start with trying to make a case for resistance to antibodies playing a role in that evolution, although infectivity is obviously also a major selective factor. And a lot of the slides I present, you can see recreated and updated several times a week at cov.lanl.gov, cov.lanl.gov. And we're still working on trying to make it user-friendly, but we've got the website going. So all of the data that I am including is from GISAID, and we have remarkably tribute to, I think, humanity, 2.4 million sequences now from all over the world, contributed by so many people as soon as they get them. So they get them out of the clinic and into the sequencing lab and into the database very quickly. So the first question I'm going to try to answer is, has antibody resistance played a role in SARS-CoV-2 evolution during the pandemic? And I strongly think it has. I have a couple of tools that we've developed at the database. And the point of these tools is in part for vaccine design. I've been working on variant vaccine design strategies. But a main feature of these tools, or a main output of these tools, is I'm part of the NIH variant committee, and there's a team of us who have sort of different lenses that we look at the evolution of the virus. And what I do is I try to capture variants that are important in spike for testing vaccine robustness and convalescent sera robustness. So I try to help people who have reagents on the lab bench, pseudoviruses, live viruses, for testing these things. And I try to capture the common forms of spike through time so they can look at these. And I have a few tools that enable doing that, and I'm not going to go over them in a lot of detail. If anybody wants to try them or to write to me about them, that would be fine. The first is called SHIVER, Historically Identified Variants in Epitope Regions. So there are two really key epitopes in the SARS spike protein for neutralizing antibodies, or epitope regions. And these are really good targets for the vaccine response. And one of them is the receptor binding domain, and it's also where ACE2 binds. So it's at the top of the spike. And then there's also the NTD region. It's called the NTD supersite, which is the N-terminal domain, and it has many epitopes as well. So what SHIVER does is it pulls out these epitope regions, and it looks for any variants that are found in any continent globally, and increasing. And if they're found temporally to have that characteristic, then if they're common enough, then they go into the database and folks can start making them and have them as reagents in their fridge, freezers, in case that form starts to expand, but also to explore the mutations that are there. Then we have another tool called X-Spike, and X-Spike looks for covariation patterns between mutations and spike and identifies the most commonly mutating forms. And here I've highlighted where the NTD and RBD is. This one is blind to whether it's an epitope region. It just finds common variants and it counts them globally, it counts them by continent. And it finds the most common natural form, both of these find the most common natural form of spike to carry a variant. And this is quite an old slide, but it gives a quick sweep of an impression that you can get. So the RBD mutations that show up repeated at this point in time are labeled in pink. There's also a lot of mutations that are showing up right next to the furin cleavage site, and the NTD mutations are labeled in green. And I'll talk a lot more about those over the next few slides. So this is now a map called an Embers map in my code. And what this is it shows you variants over time. So this is a weekly count. This happens to be a global one. And these are the variants with a Pango lineage name attached to them. And I should say, I don't track Pango lineages, I track spike variants and associated forms and then give them their Pango lineage name. So what you can see when you look at this over time, this is now the, not the counts, but the frequencies of these different forms. So it's exactly the same data, but plotted to sum to one and show the frequencies. You can see transitions where the orange is the alpha variant that came out of the UK. This is the beginning of the year. And this is the G clade, which we had just discovered last year was highly selected for G614D, which is more transmissible. This is something called the GV clade, which adds an H2V mutation. These pink guys were all positive charge at the furin loop. And then in comes the UK variant, and it really begins to overtake all of these things and shift to alpha dominant epidemic. But there are a lot of other variants that arose simultaneously, and we're all acutely aware of the South African variant, which is the lavender color here. The deep purple one came out of Brazil, and it's an unnerving variant as it was really increasing in vaccinated populations. But then here comes Delta, and it's starting to replace everything. So purple is Delta, and the bright turquoise here is just another variant of Delta, a very common one that has the same H222V mutation that was causing a shift from the G to the GV clade. Now we're getting a Delta to this sort of Delta V version that's also simultaneously expanding. So all of these complex variants are being pushed out by Delta. And another point I wanted to make while I was here is early in the year, there are a lot of one-off mutations that you could see that would start a little local spread and become stable, but then be replaced by these more complex variants. And these really tended to focus in the RBD and the NTD. And so they already started to give evidence of some traces of immune escape. So the single one-off mutations that we were seeing early in the year were either positive charge near furin, any one of these mutations, or in the RBD or some in the NTD. Okay, so how do I kind of think about this when I look at it, when I think about the evolution over the last year, that we started from an ancestral form to the G clade, and G clade just entirely replaced the ancestral form. This little H2V mutation started coming up, positive charge near furin did. Then came alpha and others that contain some resistance mutation, and then these really complex forms. I'm going to show you what they look like in just a moment. That were a lot of the variants we've been hearing about, the deep blue one from California, Brazil, South Africa, many from New York, some from Asia. So these were simultaneously coming up and beginning to spread, but alpha was increasing and pushing them out of the way until Delta came along. And now just within a few months, Delta has really come to dominate the epidemic. This is now a map by continent, so you can sort of see how things have changed. The same kind of map, but it's just continental data. Again, the first half of the year through the summer. And you can see the UK variant giving way to the Delta variant in Asia, but there were also some interesting prominent forms that were up in the spring that carried mutations in the N2D and R2D. The United Kingdom is where Delta really exploded, and they sampled so much that it became very quickly a dominant force in the global databases. So remember, turquoise and purple are both two forms of Delta, and the UK just was a pretty simple switch, and it was a very rapid switch from April to June till it became the prevalent form. Europe is following the UK. It had more diversity than the UK did, but it's certainly following the same pattern. But these continents look different. And I think they were sort of fundamentally different in their history. So this is North America. And you can see there were a lot of variants that were really prominent. There was kind of, I think this alpha UK variant had a more difficult time. It was slower to take over because there was a lot of competition between relatively fit things over here. But then Delta again emerges dominant. Africa had this beta form that came out of South Africa that was quite resistant. And South America still doesn't have much of the Delta variant. And it's dominated by Gamma and Lambda, and I'll talk a little bit more about them. But all of these intermediate strains we can learn from, I think, even though they're transitioning out, because they're very interesting. They have mutations, multiple mutations in the RBD and the NTD, and many of them carry positive charge at the furin cleavage site. So the viruses were really, if it's a green color, that means it's a mutation in the NTD. If it's a pink color, it was in the RBD, and then these blue guys are positive charge in furin. And if you just kind of let your eyes sweep down, these were the many variants we were just looking at, and it's really repetitive. And a lot of these are the same mutations showing up over and over again, or different versions of them. So E484K is probably the best known. It's a highly resistant mutation, and it shows up in many of these different backbones. Also E484Q, E484P. So you're getting a lot of convergence, and you're getting a lot of selection at these sites showing up. So if you ask, where are the mutations that occurred more than once in all of these very distinctive lineages, you see that they cluster, and they cluster just the places we've been talking about, the NGD and the RBD. So these are the mutations that happened more than one time in these little sublineages, and these are really the heart of the epitope regions. So the antibodies sit here. So to me, this is very strong evidence of some positive selection for some immunological resistance, although one could argue that these are the parts of the virus that are more able to mutate. Still, the extreme convergence and the focus in the antibody targets suggests not to me. And here's the furin loop and the furin cleavage site, which was also highly repetitively mutated. So here's kind of a more clear view of these repetitious mutations. So what are the most variable parts of spike? Well, as we were just saying, here's the RBD. There's sort of a few specific sites that really tend to mutate a lot. So I think this is probably because of functional constraints, because this region also has to interact with ACE2 to let the virus in the cell. These are more diffuse, but they're really focused on epitope regions. I'll show you that in a minute. We get a lot of mutations in the signal peptide. And then we get sort of scattered a few additional mutations throughout the proteome. But I think this also makes the case for functional domains, so that are related to selection. So the positive charge interferon, these are the patterns that you often see. And the P681R is in the delta, and people have thought that that might be responsible for its rapid expansion. I think it's certainly probably part of being responsible, but several other different lineages have also carried this R in the past and haven't gone anywhere. So it's probably not enough to act alone. And sometimes you get multiple positive charges. So it's probably going on here, we think, is the furin cleavage loop is shown right here, but the active site is shown right here and is very negative of the furin protein. And so interaction with positive charge would probably be favored. And you can do prediction scores for mutational patterns and the likelihood of cleavage. And you see that all of these would enhance cleavage. And now some of these have been shown to enhance cleavage in the laboratory. I think recombination is an important part of the evolutionary history of these viruses, and you don't have to look hard to find it. But here's just a few examples of it. So these are recombinants between different lineages. This is the whole genome now, and these are parents. So here's, for example, a P1 parent, gamma, and a B117 alpha parent. And when they recombine, this is the child, the recombinant, and it carries some red and some blue. So it's getting this base from this one, this base from that one, and you can see recombination breakpoints, and this was highly statistically significant. So you don't have to look too hard to find these kinds of recombinants. Here's one that's a little bit unnerving between kappa and delta, and it takes the 484Q that sits in kappa, which is highly resistant to zero, and it places it in a delta backbone. So recombinant may be able to enhance resistance and infectivity, and it certainly can make reconstruction of the trees a little bit confusing. This is a really simple example I showed just to try to illustrate how it can impact a tree. So this is an early case of recombination we found. And here's parent 1, parent 2, and a recombination breakpoint. There was just one in this. So this half of the genome came from parent 1, this half of the genome came from parent 2. And this was in the Netherlands. So these were all the sequences that we had locally from the Netherlands at the time we did this tree. And here's what happens if you make a tree with the recombinant. It looks like it's ancestral to this clade. It looks like it's an older sequence than these others. But if you made a tree of this half of the genome, you can see that it sits right here, identical to a lot of red sequences. If you make a tree from this half, it's sitting right here, identical to a lot of blue. So this is an artifact of recombination and not a real placement, and it can confuse things like phylogenetic and timing estimates when you have that sort of thing. Another artifact that recombination can cause is really long branch lengths. And this is another very clear example from earlier in the pandemic. But in the summer of 2020, there was a lot, like 8,000 sequences that came out of Victoria, Australia, all at the same time, big bolus of sequences, and they all have this substitution, S477N. And all of these sequences are from Australia that are labeled blue, and they all have this substitution. They were all sampled in this time frame. So we went from not having this, the S to the N form very, very quickly. These are all S, these are all N, was just a very fast transition. And these also came from the same place. They carried the same mutation. They're in different backbones. And when we looked carefully, we could show that each of these was a recombinant with another Australian sequence, wasn't happening de novo. And what you can see is they give really long branch lengths from their clade, so that can also be a little bit confounding in a phylogenetic tree. It's not hard to find recombination events when you look hard. So we recently published a paper just showing for in South Africa, when the beta B1.351 started to come into South Africa, there was a lot of different forms co-circulating, and we could very easily spot recombinants. And I just spent the weekend looking at some recombinants in Texas from Kappa and Kappa 2. So I'm from Kappa and Delta. Okay, so that's an important part of the evolutionary processes of really all of the coronaviruses. It's part of how they evolve. They also evolve by insertion and deletion. And so these are examples of deleted regions that are really favored for deletion in the SARS-CoV-2 spike protein. And here's some regions that have different insertions in them. We're also seeing some near the furin cleavage site, but this is where they're showing up, and they're showing up independently in many lineages. So like the 6970 deletion is part of the alpha lineage, but it shows up independently in all of these different lineages throughout the tree. So it's a high degree of convergence, probably. But it also could be partly contributed to by recombination, that it's happening so much and so often. It's a 6-base deletion, and it's always this way. It spans 3 codons, and so what happens if you push the C over here is you retain the I, and you have this 2 amino acid deletion. So this kind of thing is recurrent and convergent. This is actually a mapping that came from Erica Ullman Sapphire's data, and I was very pleased that she shared this with me. But she's got footprints of major antibody classes in the RBD and in the NTD and SARS, and she's working on getting these published. So these are classes that that actually are competition classes, and then she gets structural footprints. So there are 3 different classes in the NTD. They're here, and there are 7 in the RBD that she defined. And this is where they sit on the spike protein. And these are entropy plots. So you can see that the high entropy positions tend to be right on these footprints. And this is also true within these positions, although these 2 RBDs don't really have a lot of diversity in them. If you look at the deletions and you kind of blow this up, you can see that common deletions are also really focused in these footprints. So I think this is part of the regulatory mechanism for the level of resistance to different antibodies. Okay, so now we're moving towards a Delta epidemic, and this is last month's data. And these are all of the different variants that we're following, and variants of variants. Delta, again, is purple and turquoise like before, and this is the alpha variant. And so you can see that it's already shifted here, and it's on its way in the US. And one of the questions I got asked a lot when Delta first started coming out was, is it faster than D614G? And in fact, I actually think they're rather comparable. It's a lot faster than the switch to alpha. So if you look, these are on the same timeline. This is 6 months in 2020. This is 6 months in 2021. And this is going from ancestral to the G clade, which carried G614D. And you can see this extremely rapid transition globally. This rapid transition was reflected in virtually every state and city and county I looked at, very, very swift transitions from one form to the other. Whereas the switch to orange, the switch to the alpha, was a much slower process globally. And the switch to Delta was extremely fast in this time period and continues to be extremely fast as you go on. Once it's introduced, it seems to move quickly. Delta is concerning for a lot of reasons, and we were just chatting about them at the beginning of the call, but it has increased resistance to vaccine sera and convalescent sera, and it wasn't particularly outstanding in terms of vaccine sera, but in the convalescent sera, in this study by Liu et al., they were seeing that quite a drop depending on which virus the convalescent sera had been induced by. So what was the infecting virus? If it was B3551, here's the Delta, you can see that the beta Delta's worse than the other variants. Delta's worse than the other variants if it's a P1 sera, the one from Brazil. The alpha variant, it's about the same, not so bad there. And if it was just the early pandemic sera, it's not a big change. But the infecting strain can also impact the cross-reactivity of the sera, and Delta has some trouble in that light. It also is maybe about 4-fold less potent in terms, or less sensitive in terms of vaccine sera. And it requires two vaccinations for good potency. And that's different. Some of the older variants didn't. Associated with higher viral loads in infection, this is really an extreme number, but even if it's just partly true, it's pretty impressive, 1,000-fold higher viral loads during acute infection. So this could also be part of the immune evasion response. And Delta variants can break through vaccine protection. We're seeing more and more of that. So if you want to walk through sort of a history of this, here's October, November 2020, a global map. And what you can see is the G and the GV forms. These are precisely the G and GV in spike. Alpha was just entering the world, and all these gray guys are single, one-off or two-off mutations of G and GV that never really stabilized. So this is what the world looked like at the end of 2020. We were seeing alpha, we were seeing the Zeta coming into Brazil, Beta coming into South Africa, and lots of positive charge and furin all over the world, just single mutations. And 2 months later, if you look at it, we really had extremely rapid growth of the alpha variant. So it was really taking over. We're beginning to see diversification in the US, really pretty extreme diversification. A new form had entered Brazil, which we call Gamma, which is quite resistant. And the beta form in South Africa is really beginning to take over there. So this is now the next 2 months. And you can see that the situations got very complex in the US. We had many variants rising in Mexico. Also in Asia, where we had sampling, we could see variants. This new Kappa form was showing up in India and concerning. And we were seeing a new form called Lambda in Chile. So this was a very highly mutated form that was coming up in a very highly vaccinated population. Then we get to late spring, and really the alpha is dominating everywhere, but Delta is starting to take over in England, and purple and blue are both Delta, 2 different variants. So finally, here we are now, and you can really see that Delta has become prominent in most of the world, not yet South America, but probably soon to be so, given the rest of the history. And here's just an expanded view of Europe, because England samples so much, these circles are related to sample size, that it just swamps out, the UK swamps out everything else. So I blew up Europe so we could actually see it, but you can just see April, May, June, and July, very rapid shift. And again, in the US, May, June, July, a rapid shift towards purple and turquoise. And all of these many variants that had arisen didn't survive, but I think are interesting to study anyway for the reasons I just explained to you. You can see a lot of presumed resistance mutations and a lot of known resistance mutations from the laboratory in these variants. But anyway, we're also switching towards a Delta. And these are the 4 different forms of Delta I'm tracking. Cases, severe cases and deaths are increasing. So if I can't see my whole screen, unfortunately, but here, yeah, this is the United Kingdom. And you can see that's really coming up here. We've also got Freedom Day, a societal experiment that we'll see the results of over the next month, I think. Then we've got Israel is coming back up, and we've got the United States coming back up in cases. And you can see the parallel in those 3 countries of Delta, and Delta just came in super quickly in Israel just the past couple of weeks. And the really bad news out of Israel has also been the past couple of weeks, which may be coincidence, but it is a coincidence in time. So it also might be something more. Here's that Brazilian P1 variant. And you can see it was actually coming into prominence and holding its own against the Alpha UK variant, but Delta has swamped both of these out. And here's another form of Delta that's just coming up in the US that's called AY2. In Pango lineage terms, it's got more mutations. So we can follow all of these variants in Delta. So this is just the maps that I've been showing you where Delta looks like this. It's just these 2 forms. But here now, these are all the common forms of Delta globally. And so we can begin to ask the question, if you compare a new mutant form of Delta, say that has P251L, which is this form, or this is a very common form, it doesn't have the I95 mutation. Are any of these favored over others, or are they just founder effects that we have different distributions here? And so far, I think I have only evidence of founder effects. So here's the United States. And I also track the resistant forms that I'm nervous about in terms of vaccine resistance. So alpha, beta, lambda, and this one is actually gamma. We're all coming in and we're beginning to replace alpha anyway. And then Delta enters the picture. And this is the first form of Delta into the United States, this little brown one. But these are all forms of Delta. And then here's AY1 and AY2, which carry an additional K417M mutation, both, but they also both carry additional mutations in the RBD and are more complex. So anyway, this one comes in first, and this one is really dominant. But you still see these other forms all present. You don't see anyone that really stands out. Only these 2 that have just entered are maybe coming up pretty quickly. So we've got to keep an eye on them over the next few months. They've only been in the world for about a month that we've been sampling them. So these are going to be worth tracking very carefully and following. And certainly, once we see something like this, these get made in the laboratory. And that's the good news. So we've got them. And that takes maybe 4 weeks to get the order in, and then another 2 or 3 weeks to test it. So we try to test things in the early phase. So if something does become dominant and important later, it's there and we know about its mutational profile. Where AY1 is common, it's not really dominating yet. So this bright purple line is AY1, but this brown, simpler form of Delta is what's strongest. And these next couple of slides, I'll just show that in Portugal, AY1 is common, and in Switzerland, in Geneva in particular, it's common. But I think so far we're mostly just seeing founder effects driving whatever form of Delta is at the endpoint. And AY2 is our US variant. It's this lighter pink guy. And it comes up initially, but then it doesn't really seem to take over yet. So we just have to monitor it longer. I don't think we can say yet if it's going to be more prevalent soon. Anyway, I do this a lot. These are horrible plots to look at, actually, but I find them fascinating, so I enjoy looking at them. But they go by continent now, and they look at all of the common Delta variants. I also look at these by something called Isotonic Regression, which we came up with as a way to look at the D614G mutation. And what it does is it probes oligosate, and it finds every country on the planet, or every city or county, you can go at different levels, where there's enough data to see statistically a transition between one form and another. So this is an example of alpha and alpha with 490S, which I think is maybe an important resistance mutation. So when I'm comparing is now alpha and alpha 490S directly, because alpha, we know it was more transmissible than earlier forms. So I want to know if this is more transmissible than alpha in places that they're co-circulating. So these are all the places they're co-circulating in the world. And they were all found to be significantly increasing, every single one of them. And this is what significant increase looks like. This is the case from the Netherlands, which is in this list. It's right here. So if you looked in March, you'd see that there was no 490S. And what these are, every dot represents a day, and the size of the dot represents the number of samples that came in from the Netherlands that day. And this is the fraction of alpha that's 490S alpha. So there was no 490S, and all of a sudden you start getting a little when you sample, and you get increasing frequencies as you go over time, and this is statistically significant for this country. So when I see a whole lot of places like this that are always going one direction, I think that this form is actually got a selective advantage over this form. So it's, this consistency of pattern. And when I look for decreases, I don't see decreases on the other hand. So I think there was 1 out of 15 that had a decrease. So that's very suggestive that it's positive selection to me, this recurrence. But here's another example where I don't think we have positive selection. It's that H2V in Delta that I've been showing you, the bright turquoise band that comes up above the purple Delta in all those maps. It's very common, but I think it's a founder effect. It was found in 21 countries with enough data co-circulating to enable assessment of whether this was increasing relative to Delta. And in 7 countries it increased, in 7 countries it decreased, in 7 countries no change. And you can also get an increase due to founder effects and sampling biases. And the fact that this is randomly distributed, unlike the other, which was 15 out of 15 increase, suggests to me that that we don't have any evidence yet that this is more fit. But it's very common, and we're gonna study it anyway, just because it's so common, and it doesn't hurt to have it in the panels. Here's a quick view of Israel, again, because this is a place with 85% vaccine, double Pfizer vaccines, so fully vaccinated population, where they've been having a recent surge in infections and also hospitalizations and deaths. They're beginning to get deaths again. They had no deaths for a while. And you can see Delta has really come up very suddenly in this same time frame. And the Delta variants in Israel, here are all the specific Delta variants in tracking. And here are the other major strains of importance. In the spring, as they were vaccinating, actually we were seeing a higher level of Bette from South Africa and Lambda from Chile entered the Israeli population. But all of these things are giving way to these forms of Delta. And these are kind of generic forms of Delta. You can also start to look at specifically what other mutations show up. So in the last 60 days of sampling, these are all the variants out of Israel. And the purple ones are deltas, the orange ones are alphas, the black ones are something else. And you can see that there's certain mutations that are showing up that are potentially very interesting in, say, the alphas, 494P, 490S and 490S in 2 different forms. So these are potential resistance mutations that are normally quite rare, but they show up in the backbone in this vaccinated population. Chile still doesn't have Delta, and it was highly vaccinated even in early spring, and we started seeing the emergence of this form that's called Lambda by the WHO, and it's now giving way to this form that came out of Brazil that we call Gamma. So what you can see here is that the UK variant never really got much of a toehold, but that these 2 forms were very prevalent in these highly vaccinated population. We're kind of just trying to keep an eye on the US, and we have regions that are very low vaccination rates and regions that have quite high vaccination rates. And I was looking to see if there's any differences that I could see in the distributions. And this is just a hypothesis raising exploratory thing. The one thing I could see is that earlier in the spring, the more highly vaccinated populations really tended to get a higher accumulation of this, this gamma form out of Brazil that's highly mutated. And this was actually happening sort of all over the world, but everything is giving way to Delta. And here is the AY2 variant that's coming up of the AY2 Delta variant. This is the H2V Delta, and this is the generic Delta. So there's sometimes difficulty in calling these things, and I just want to emphasize that a variant isn't one thing, it's an evolving lineage, and the data can sometimes be a little bit rough and take a while to settle down in sequencing. And they change names. Pango gives new designations over time. So the AY3 is pretty new, it's about a week old. So all of these things have been just called B.1.6.7.2, and suddenly they became called AY3, and another set on July 15th, they changed their names. Then a whole bunch of new AY3s were added. So these kinds of names are not in stone, they're transient, and depend upon the tree at a given time. And sometimes it can be even difficult to see why they call a new lineage. So here's a basic B.617.2 map of where all the mutations are in spike. And these lines are the shared mutations, the little dots are the random mutations that are showing up in the Delta lineage. And some things, there's like little sub lineages within it. The H2V, for example, shows up. The AY3 spike has a very, very similar pattern. All of the consistent ones are shared. All the broken ones are shared where they're not in all of the variants. So these are 5,000 variants, these are 1,500 variants. Every row is a different sequence, and this is position in spike. And you can see that the variation is actually recapitulated in this new Pango sublineage. These are the other Pango sublineages of Delta, and they are much more distinctive. They have things that exist very consistently and only in those viruses, like the 417N is found in these 2, but not in these 2. This one almost always has H2V. This one almost always has 95I and 258L. So they have distinguishing characteristics in spike. But part of the problem with trying to call these things early on is that sequencers have difficulty getting them when they're new variants. And so all this black area is unsequenced. It's all just gaps in the sequence. So you might think that this particular AY3 didn't have this mutation, but it turns out that every time it's missing, it's a gap in the sequence, and it really does have this mutation quite consistently. So you have to be a little bit careful about having in mind these things. And all of the Delta sublineages have these problems. They have a lot of gappy sequences in them so far. So I think, these are still resolving. You can have situations where people call this sort of thing differently, and it worries me a little bit. So there's a very consistent deletion in all of Delta. It's a deletion of 156 and 157 in our terminology, and a change to a G. So it's six bases are deleted, and again, it's across 3 codons. So you have to repair it. You either have to push the G and A over here, or you have to push it over here. And when that happens, you get a G, and it's brand new. So we align it to the R, others might align it to the E. And there's been a lot of sequences out that don't have the G that people are making in the laboratory, but the G always occurs in nature. And these things can be kind of tricky. So sometimes there's ambiguities and confusion, and the sequences are being propagated to represent a different variant like Delta. we always put this G in, but a lot of people, I think, have been making it without the G in because of this confusion. So just sort of a little warning. Also, Pango lineages aren't always really obvious lineages. Like here's one called B.1.526, and it doesn't share any spike mutations with all of the other B.1.526s. So it's a very distinctive subset. This is now 1,700 sequences. And it's also quite distinctive in the other parts of the genome. So I'm not sure why this was called a lineage, but it means that two extremely different spikes are grouped in that same group. So these things are a little bit soft. They change over time and they're not always clear. I'm just going to show you one example of the other kind of thing besides algorithmic stuff that I do to pick out new variants. I actually try to keep an eye on other issues. Like, for example, Botswana is a hotspot. The New York Times said this week that it's one of the highest transmission rates in the world. And as it turns out, Botswana is not any lineage I'm tracking. It's distinctive. I don't have it. I have gray for unknown. And so, look into it. What is it? Well, it's actually a form of kappa. It's a distinctive form of kappa that I would miss by my tracking tools. And so that becomes an interesting one. It doesn't mean we need to panic and say, oh my gosh, there's something out of Botswana that's really dangerous. But what it would be nice to do is get these ordered. So, we have them to test just in case this one starts to expand. So that's the kind of thing we're trying to do to stay ahead of the virus, as an example. So in conclusion, lineages that had local geographic expansions in 2021 really very often carried mutations in both the NTD supersite and the RBD. They also often had positive charge interferon. So a lot of these mutations we know from laboratory studies on the bench confer relative resistance, not complete resistance often, but relative resistance to different monoclonal antibodies, vaccine sera, and convalescent sera. So these do have functional consequences, and it's so striking to me that both were carried that I feel like this has got to be a trace of resistance that we're seeing on the fitness of the surviving viruses. And this positive charge interferon cleavage, the same kind of thing. I think experimentalists are really more interested in the RBD and not so much the NTD, typically, not everybody, but I think that the virus is giving weight to both of these by the probable emergence of resistance to both of these simultaneously. Mutations are very often recurrent in these lineages, and they're really focused within key epitope regions. And some of them may also impact infectivity, especially RBD mutations can do both, but also NTD can. Usually convalescent sera is more severely impacted by resistance than vaccine sera, which tends to have more potent neutralizing antibodies, so able to cope better with resistance mutations, but this could still be a selective force. And we are still learning how variant infections impact convalescent sera. So I showed you that one example of if you're infected with different strains, your target other variants. They have a distribution of how well they're able to target other variants. So it's not just a simple story. The ongoing transition to Delta is really rapid, and of course it's continuing to evolve. The last time we counted, we found there were about 2,500 different forms of spike that are all in that Delta lineage. And about 150 of those were found more than 10 times, so at least they've been transmitting a little bit. So trying to keep an eye on this and figure out what's the best, most central thing for reagents for study is tricky. And there can be some issues where people come up with different versions of Delta. And one thing we're not doing in the field very much is saying which version of Delta we're using. And I think or any variant. So I think we need to start being much more precise about that, because there's only usually 9 or 10 amino acids involved. And if people call Delta with 3 or 4 amino acids different, it might give a very different phenotype, not just one thing. I think the knowledge base from all the previous variants is informative regarding both infectivity and resistance mutation. And some of them are already beginning to be newly resampled in the Delta context. And more convergence is likely. So understanding these variants that came up before will be helpful, I think, for understanding what we need to worry about in the future. And I'm hoping that the convergence we've seen in the past will enable more robust variant vaccine designs. We don't know, how long Delta will last as the predominant form. We don't know if the next virus out will come from Delta or from an older variant or very distinctive variant that's not part of the Delta lineage. But we are beginning to learn what convergent mutations are that can confer relative levels of resistance. So perhaps these can be incorporated into vaccine design. And in fact, indeed, that's what we're trying to do. And this is basically saying the thing I just said. The next sweep may come from a lineage unrelated to Delta. And a way to think about that is we were having a slower transition to Alpha, but a really major transition to Alpha. And yet Delta didn't build on Alpha. It came from a completely different pathway. So sort of trying to keep an eye on the current complexity of the virus, where it's going, and what these single mutations do is all going to be an important part of our success in the future as we, advance second and third generation vaccines. And hopefully we will be successful. I think that is yet to unfold as well. So finally, I just want to thank my funders. Los Alamos National Lab funded most of this work, but I have a little bit of funding from the NIH and from the Bill and Melinda Gates Foundation. And I do almost all of this with my husband, who's retired. And so his 401k is supplying a lot of the funds for this project. So anyway, that's it. And I'm happy to take questions if there's time.
Great. Thank you, Betty. That was amazing. There are some questions in the chat and in the Q&A. Sam Bazzetti asked one, what are the implications of rapid replication of Delta in partially vaccinated populations?
In partially vaccinated populations. So I actually think, I think that's a real problem, because what you have, or even in fully vaccinated, because Delta is able to get in a little bit, right? So say me, I've got wonderful antibodies because I have the Moderna vaccine. But if I was to get infected with Delta, I would be a perfect selective environment for Delta to learn how to evade vaccine antibodies. Now multiply me by hundreds and thousands or millions, and the evolutionary space this virus has to develop in. So I think if that's what you were getting at, I think it can be a severe problem. And partially vaccinated people will have vaccine antibodies, but wimpier ones, which is an ideal selective environment for Delta. So I think, or any variant, right? So I guess I feel passionately that we made a big mistake about the masks. I feel like that masks in public places is really our other big tool, right, for preventing infection. And the other catch with this whole thing is a lot of our population can't be vaccinated, the kids and the teenagers, and Delta seems to be hitting young people more often. So the masks can not only protect you by wearing the mask, but even better, they protect the person you're breathing on in the same space. So we're protecting our communities by wearing masks. And I feel like I want to limit this virus's evolutionary space as well as we can. Maybe it's futile, but I wish that we were encouraging masks indoors very strongly now, and I think we should be, and unfortunately we're not. That's my opinion, and I think that it's related to that question. I'm sorry, I went off on a tangent a little, but I think it has bearing on the question.
Thanks, Betty. Some more questions have come in. Stuart Ray asks, well, first, Betty, this is always, as always with your work, informative, dense, elegant, and illuminating. Thank you for your global perspective. Your analysis emphasizes the amino acid states, which are familiar and obviously potentially functionally important. But I wonder if lineages and recombination segments in particular might be more robustly recognized by synonymous sites that might be less subject to convergent evolution. Is that something you've looked at explicitly?
I haven't looked at it. No, it's a very good question. Well, of course, it's a good question, Stuart. but I'll tell you one thing about. I focus mostly on spike, and then I extend to the other parts of the genome when I want to look for recombination or something. so many of the lineage mutations are non-synonymous, just really a high number of them. So it's a really big proportion, and that's also suggestive of a positive selection. And I haven't systematically looked at that either, but it's really a lot. I am positive because I do the silent and the non-silent, and I do maps and stuff. I haven't done it mathematically and been very quantitative about it yet. But I think the fact that you can see the recombination involves silent and non-silent mutations and messes up lineages, right? I mean, the whole point of a lineage, right, whether it's silent or non-silent, it's going to break parts of the genome and give them different phylogenetic histories. So I don't know if I answered the question well, but
I think you did. Maureen Holtland asked a question. Are you tracking non-spike changes like NSP1 variation, as well as what you've been doing with spike?
very, very little. And I wish I could do more. It's a bandwidth issue. Much of the work that you've just seen has been done by only 4 people. And my husband and I have done the bulk of it. And so we just don't have the bandwidth. And I'm very focused on the vaccines, partly to help for reagent design, but also partly for vaccine design. And we've got 3 in the pipeline that I'm hoping will help with variants right now that are being tested by Drew Weissman and Dan Baruch. And many others are obviously looking at this kind of question too. But what I do is when I have a variant in spike, I track, my colleague Will Fisher creates for me a full-length genome that's most representative of that variant. And then we do, create alignments of these very, very representative sequences. So we can sort of track them that way. And you do see convergence in other parts of the genome as well, where you're seeing repeated mutations that maybe have a lot to do with expression levels and infectivity. And I just don't have time for that question, but God, I would love to look at it.
Great, Betty, thank you. There's one last question from Greg Forrest. What do we know about these spike epitope mutations, the impact of these mutations on the affinities to receptors on epithelial cells? My group models how infections spread in the lung, and this is a critical issue for us.
It's a really great question, and I'm sorry, I just really don't know the answer to that question, but it's a profoundly important question. I'm not sure.
Great. Again, Betty, thank you so much for presenting to all of us. It's been remarkably informative and a fantastic talk, like you always do.
Thank you for inviting me. It's really nice to be with you guys today.
Joel, do you want to close us up?
Hey, no, thank you. This was really a fascinating talk and really very exciting. Let me actually just ask one last question.
Okay.
I happen to have the floor, and that is, what insights do you have about mixing vaccines, particularly using the vaccines that use different mechanisms? Do you see that as being potentially useful in reducing viral evolution?
I do. I'm actually quite hopeful about that, both from an economic and practical point of view, but also from like you said, the triggering of different aspects of the immune response. But I guess that story is just unfolding now, and it's going to be maybe helpful. And I think that it's really important to sort of study carefully before we start implementing. But I think there's hope in that perspective. I'm a T-cell believer as well as an antibody believer. I've only been talking about antibodies, but the idea that the T-cells are really helping us stay healthy after we've been vaccinated, even when we're infected, it seems very to be panning out and becoming there's stronger and stronger evidence for it. So a really good complementary vaccine that's able to tap into both well, and maybe it starts looking out of spike as well, but other proteins that make particularly beneficial T-cell responses or rigorous T-cell responses, I guess, would be another route for the the future, hopefully the near future.
Yeah, no, thank you. And I have to say that as an ex-medical student and clinical pathologist, the early popular reporting of the pandemic was weirdly antibody oriented. And
Yeah,
it was as if T cells had been abolished. Nobody discovered them. And then about 6-8 months ago, reporters discovered T cells. They said, hey guys, they're T cells. Amazing. Anyway, interesting. So wonderful talking.
thank
I
you.
think we're going to have to wrap things up now, so we'll move the rest of the agenda to future sessions. And I really apologize for that, but this was a fantastic talk and
Thank
really
you.
was what we needed. Okay, thanks everybody. And I think with 1 minute we should just wrap it up. Thanks.
Bye now.
Bye bye.